Welcome to the Duck Dive. Today we're looking at a paper from 2008, the Raj paper, that really kicked off modern quantitative RNA imaging.
It really did. And the core idea is uh, almost backwards. It's
Huh.
so counterintuitive.
Right. To get a brighter, clearer signal from a single molecule, their big idea was to use less labeled probes.
Dozens of individually dim probes instead of a few that were supposed to be super bright.
It sounds crazy.
It was a simple pivot, chemically speaking, but the impact was just enormous. Our mission for this deep dive is to unpack how that one shift made this technique, smSAH, accessible for, everyday biology.
So before this paper, what was the big challenge? What were people trying to do?
Well, the goal was to count mRNA molecules, and I mean get actual integer counts, 1, 2, 3.
In a single cell, not just in a soup of cells.
In a single cell, exactly. And crucially, to know where in the cell they were.
And the old method from the late 90s was kind of a brute force approach.
Totally. The thinking was more is better. Take a few long probes and just cram 5, maybe more fluorescent tags onto each one.
But that creates its own problems, doesn't it?
Oh, huge problems. The synthesis and purification alone were a nightmare. But the bigger issue was self-quenching.
Right. If you pack the fluorescent molecules too close, they basically just cancel each other out.
Exactly. So
Yeah.
the signal became super unreliable. The spots would have wildly different brightness levels, and you couldn't tell a real signal from, just background noise.
So the new method flips that on its head.
Completely. Use 48 or more short probes, but each one only has a single fluorophore. You avoid the quenching, and everything becomes simpler.
Strength in numbers.
Yeah.
Okay, so let's get into the results. Figure 1 seems to be the big validation step here.
It is. The fundamental question was, is this little dot of light we see, which is made from 48 different probes landing on one target, is that really one single molecule of mRNA?
How do you even prove that?
You need control. So they engineered these cells to make a GFP mRNA that had a special known sequence, a repeat at its tail end.
The 3-foot UTR.
Exactly. So they had their new probes, the 48 tiled ones, targeting the main body, the GFP message. And then they used an established, trusted probe that only targeted that special repeat sequence.
So if it's one molecule, both sets of probes should land on it, and you should see both colors in the same spot.
And that's exactly what they saw. The colocalization was amazing. 81% of the new GFP spots matched the control spots.
And the other way around.
85%. I mean, it was basically a near-perfect overlap that confirmed it. Each diffraction-limited spot is one intact single mRNA molecule.
Okay, so they've proved a spot is a single molecule. That's a huge step. But I guess the next question for you, the listener, is what's missing? What do you need before this becomes a go-to tool for every biologist? You need to know it's quantitative, that the numbers mean something.
Exactly. Can it actually track real biological changes? That brings us to figure 2.
Where they tried to do multiplexing.
Right. Tracking multiple genes at once. They used 3 different colors to look at 3 different genes in human cells. Then they treated the cells with a steroid that they knew would change those genes' expression.
And the proof is in comparing it to the gold standard, right?
Yes. They measured the RNA levels with the traditional method, quantitative RT-PCR, and then with their new smFISH method.
And did they match?
Perfectly.
Huh.
The genes that RT-PCR said went up, like FTBP5, they saw more dots for in the microscope. The genes that went down, they saw fewer dots. It proved the technique was specific, sensitive, and, most importantly, quantitative.
And it wasn't just for cells in a dish. Figure 3 shows the versatility.
Totally. This is where you see the real power. They showed it works in whole organisms, in complex tissues.
Like in the C. elegans worm?
Yeah, they could see the L2 mRNA was only in the gut cells, exactly where it should be. Or in a fruit fly wing disc, they saw the DPP gene expressed in this beautiful sharp stripe. It preserves all that crucial spatial information.
Which brings us back to that core conceptual innovation. It's not just a better recipe, it's a whole new way of thinking.
It is. You're moving from this fragile, difficult chemistry to a statistical principle. The signal comes from the fact that you need dozens of these little independent probes to all land in the same tiny spot.
So the specificity is built in.
It's built in. And because it was simpler and they released the software to design the probes, any lab could start doing it. It just opened the floodgates for studying cell-to-cell variability.
And it's the foundation for what's happening now, right?
Yeah.
With methods like MERFISH.
Absolutely. That tiling logic is the direct ancestor of the technologies we have today that can image hundreds, even thousands of genes at once in a single sample.
Okay, so to wrap this up, let's nail down the 3 big takeaways. First, there's the chemical simplicity. Using lots of short, singly labeled probes just works better. It avoids all the old problems.
No more self-quenching. It's a much cleaner, more robust system,
Second, it's quantitative. The single molecule counts you get actually match up with the bulk gold standard measurements like RT-PCR.
which is critical for the method to be trusted and widely adopted.
And third, the spatial scope. It works in multiplex, and it preserves location in complex developing organisms like worms and flies.
A really powerful technique. But it does have one key limitation we should mention.
What is this?
It requires fixed specimens. The cells are dead. You're looking at a snapshot in time. So a final thought for you to mull over. If the next great leap is to watch these processes happening live, what kind of technical breakthrough would we need to turn this fixed snapshot into a real-time movie of transcription inside a living cell?
Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. 1. To help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2. To build a supportive community where we can exchange the most valuable experience-based research advice. 3. To share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.